What's up, bro? Oh, you're in.
What's going on, my
Are we
man?
doing this?
We're
All
doing
right.
it.
All right, let's do it, man. How's it going? How's your day?
Literally just got back from up later than I wanted. Just got back from doing a leg workout. Only got to do quads, but it is what it is. The posterior chain can wait until another day. How about you, my man?
Doing pretty good, man. Just finishing some client stuff. Did you do squats, barbell squats?
God, no, my femurs are so freaking long. I got to do all
I
the
know
heel elevated, weird. I got the giraffe legs.
that's why
So
I'm
short torso,
bugging you
long
about it.
legs.
Yeah.
Yeah. So I did some heel elevated goblet squats. I like that one a lot. Did actually like starting with leg extension, kind of warms up my knees a bit. And then two different machines. But yeah, it was good. Yeah, if everyone watching now, we're completely just winging
Winging it.
winging
We
it.
have no plan.
We have no plans at all. So probably just got I don't know. I think we should each chat a little bit what we're up to
Yeah.
nowadays individually, and then we could kind of just see where things go. We'll be going off some
Pull up some
stuff you guys are
topics.
maybe asking and just really trying to start with the elementary discussion of these things, but slowly try to work our way up to the higher level stuff. And hopefully some of you guys that maybe wouldn't have understood these concepts initially, hopefully we could slowly work that up and make you guys think critically about some things.
Or just have some fun.
Yeah, that too. That
Yeah.
too. I'm literally making our thumbnails we speak right now too, so people know.
Gotcha. So I think a good place to start is actually that recent story that I posted about with the circadian biology affecting pancreatic cancer. And then you mentioned one of the genes that I actually did not hear about before. I think it's like a acronym that is pretty obvious. Do you remember which one
Was
it
it
was?
clock? Was it clock
There we
gene?
go. There
Yeah.
we go. What is that about? I actually have not heard of that one. And what does it do? What's the role?
Yeah. So it's a transcription factor. So transcription factors, guys, those are what ultimately bind to DNA. So cells respond to stimuli. Let's say, you can tell it's daytime or nighttime. Your cells sense that on the surface. And all the way at the end of that intracellular pathway inside of the cells, all these proteins working together, transcription factors would actually bind the DNA and let the cells respond. Cells respond by expressing specific genes. So, good example everyone's familiar with is vitamin D. So, you get sunlight, your cells can sense that signaling from the vitamin D. It goes inside and makes these cells express certain things, anti-inflammatory, better mood, etc. This regulates, it's called the clock gene because it's the master regulator of the day and night cycles inside of all of our
Mm,
cells. So that regulates a lot of different things, such as everyone hears about the blue light blockers, cortisol production. So you want to have at nighttime with the blue light, you get higher cortisol and lower melatonin secretion. Those are two gene targets of the clock transcription factor. So think about TikTok. The clock tells your cells if you have that blue light, that clock gene can prevent the secretion of melatonin. And that's actually something I see. I have that mutation, and I put that on my story too. People were asking me like it's some heinous, foul mutation in of itself on
mm,
its own.
mhm.
But that on its own, it just means that your cells won't be able to tell when it's daytime or nighttime. Meaning, I get my sun exposure every morning. I wear blue blockers. I limit blue light at night. I still have trouble actually falling asleep. So sublingual melatonin really changed my life. I hated the pills with the binders and stuff and
Yeah,
everything.
yeah.
But basically, that just means my cells can't really secrete the melatonin at the right time at night. It makes me much more of a
It's really interesting.
night owl. And I was more so commenting on circadian rhythm definitely plays a huge role in disease progression, cancer included. So imagine if your circadian rhythm is genetically disrupted as well. Obviously, you have the watching TV right up to your face playing video games, eating potato chips at 11 o'clock at night. That's not genetic related.
Yeah, yeah.
But it kind of piggybacks off of the lifestyle with the genes, of course.
What about the if you have, for example, already a mutation in some of the genes that process melatonin, for example, or tryptophan metabolism in specific, and then you have this, would you think that supplementing melatonin would be a good idea for people? Meaning,
Yeah, sorry. So you said if there's a tryptophan thing as well.
yes,
Yeah,
exactly.
yeah. So for you guys that aren't aware, tryptophan is the amino acid that gets converted into 5-HTP. A lot of people have heard that in the context of raving. You know, people going out, rolling, depleting their serotonin. You take 5-HTP to restore it. So you have tryptophan, 5-HTP, and that gets converted into serotonin and melatonin. So people with mutations in tryptophan synthesis, metabolism into 5-HTP, that would actually be like a double whammy with the clock gene.
Yeah.
Because from the get-go, your cells aren't going to be producing as much melatonin, period. And that will probably also bleed over into some possible low mood type things. It's interesting because a lot of these depression-related serotonin genes also affect melatonin production. So they're going to have less serotonin to begin with, so lower mood. They're also going to sleep worse for the lower melatonin. So it's kind of like a double whammy there. We know how horrible poor sleep is for our overall biology, mood, inflammation, etc. But yeah, I mean, depending on the person, sometimes the 5-HTP works really well, but I've noticed it's very easy to overshoot because, you know, there's one less enzyme, you know, I call it like a safeguard to not go over the top. And that's a good point I want to mention too. Like a lot of people, I want to make a post about, people ask me a lot about like the Ray Peat serotonin stuff and everything. I personally never really followed him that much, but I know he's had some good thoughts. And a lot of people think, you know, oh, more serotonin's bad, but you got to draw a dotted line where your cells should be at. And what we're talking about here is correcting a deficiency, not going over that. So if your serotonin and melatonin is lower to begin with, your cells are going to be craving that. So boosting up serotonin when it is actually lower than it should be in your cells, I think is beneficial. But I think elevating anything above that baseline, anything more than, you know, once or twice a week, that's where you run into those tolerance issues. That's why people take Adderall for a day and the next 2 days they're just completely wiped, because you may have a little bit of a deficiency, but you're going to be shooting way over that, especially with something like Adderall with what they're prescribing.
You know, that's funny you mentioned Adderall in specific, because I have tried it before and I've always had a negative response with like few milligram doses, never going above 10 milligrams, because I would just get extremely wired and adrenaline rushed that it wouldn't
This
help
is melatonin?
with what it's Adderall.
Oh,
Adderall.
yes.
It wouldn't help with just getting things done as it's supposed to do. And it's the complete opposite if I take other stimulants like caffeine, theobromine, etc. I get way more focused and I'm able to execute tasks a lot better. But Adderall in specific makes me worse. And I think the reason has to be because it's pushing me just a little bit beyond what's my baseline, like what should be my normal dopamine production.
And have you ever had Vyvanse before?
I haven't.
Yeah, see, that's one. I personally like that one a lot more. They're actually the same drug. I'm sure you know this, but for everyone listening, Adderall and Vyvanse are literally the same drug, just a different isomer. So a different 3D version of the same molecule. And the main difference between the two is Adderall raises dopamine and adrenaline very high. Vyvanse, the other isomer, it elevates adrenaline, norepinephrine much less. So the ratio, it's a lot more dopamine to adrenaline ratios, which
Interesting.
I love it. I think we're similar. Probably the slow COMT enzyme. You know, we're probably very higher levels of dopamine, adrenaline to begin with, and much smoother overall. And smoother overall because you're not having a lot of those most of the side effects from these ADHD medications from the adrenaline. You know, the clammy hands. You know, the jaw, the this, the that. I'm not endorsing either of these, by the way, people. You know, we're just discussing here with our experiences there. I think that there's so much other nutrients you could be taking, like you mentioned, caffeine, theobromine. And I actually know what I've been trying a lot that I've been loving. Have you ever tried Dynamine before?
No, I haven't.
Are you familiar with it?
No.
So it's a xanthine, similar to caffeine, but it has like a quarter of the half-life.
Mm-hmm.
And I personally metabolize caffeine slower than a normal person. And I started taking it. I love it for the gym. I normally have just espresso in the morning. I cut my espresso in half and added a little bit of that. Much better, like tenacity in the gym. And I think more of a nootropic effect versus just caffeine. I mean, nothing, you know, earth shattering, but just subtle differences there. And I noticed I've been able to get to sleep easier with it. And then on the other side, they have what's it called? Theacrine or teacrine.
Yeah, that one.
Where they have
that one I've tried before.
like that one?
Oh, so good. It's a clean focus, and it's different than just pure L-theanine. I think it has to be where they
No, I'm saying
pull it from.
teocrine, the xanthine. Yeah. You're talking about. I'm talking about teocrine, not theanine. Like the
No, yeah, theocrine itself. Isn't it? Let me pull it up really quick. Could have sworn it was a type of theanine, but yeah, keep on going.
But yeah, that's basically a longer duration. Caffeine and theobromine better than pharma drugs. Yeah, I used to take these caffeine pills that had green coffee bean caffeine and the theobromine. I really, really like it. Especially, I really noticed the vasodilation effects. Like I don't know, definitely feel dopaminergic from the theobromine. I respond well to that one as well.
You're right. It's a xanthine. It's nothing to do with theanine itself. They call it theocrine. Never mind.
Oh, there's someone said I put questions in the questions box. Is that a thing?
I had no idea. Yeah, they asked some questions.
Oh, you have that on your end, I guess, because you're the host?
I see. All right, we'll go over them afterwards.
Yeah.
We'll spend like 30 minutes on questions. Back on
I
that
didn't know there was a question box. That's news to me. It's good they finally have that, so they're not buried in the chat.
Back on that serotonin topic, one of the things that I found just the past week is the production of serotonin in the enterochromatin cells, so in the gut itself. That's usually what's associated with the really negative side effects of serotonin, because it causes contraction of the intestines, and to eliminate things, it basically mimics the symptoms of IBD. It's producing a lot of 5-hydroxytryptamine. It targets the immune cells in return, and they promote this pro-inflammatory cascade that then causes IBD-related symptoms like Crohn's and ulcerative colitis. So it seems to be that where it's located, the tissue is also important, not just the receptor type, not just how much serotonin is produced, but tissue-specific effects can be different.
That's one thing that's always really fascinated me, was just the tissue, like organotropism. So the tissue specificity of how do certain cell types require higher levels of certain nutrients, and how does the body and all these transport proteins, how do they know
Manage that.
where to obviously they know where to go, but you would think it would be kind of roughly the same. How do they know preferentially to bring more, CoQ10 to the heart, or more this or more that? I know we don't have the answer here, but just so fascinating to me how they do that. The first thing I think of is, I think a lot of this stuff is transported by, there's transport proteins, and there's also the exosomal stuff, like the vesicles and how you could traffic them specifically. The whole drug delivery side is super, super interesting. I wanted to go in that, knowing like extracellular vesicle biology stuff, like drug design.
Yeah.
But really, really fascinating how they know how to go and which. I swear, individual cells are smarter than the person they make up. I have no idea how all these cells know how to do these. I mean, how many reactions are happening in one cell within a minute? Like what? Hundreds, thousands, tens of thousands?
There was a paper that I remember reading where it says every 15 seconds, all of the proteins inside of a cell have collided with every other protein inside of the cell.
15 seconds?
Something like that. It's definitely within seconds range. And considering how many proteins there are millions of proteins inside of a cell at any given moment in time, forming, not forming, being broken down by ubiquitinases. So it's a lot of molecules interacting with one another within a couple of seconds time. It's very interesting how it's all coordinated. Very, very specific too.
Now, what are you drinking now? Tea?
Yep, some jasmine tea. It's pretty good.
Yeah, and I was wondering what your thoughts were too on the what was the paper that came out? I'm drawing a complete blank right now.
The Sinclair paper, I'm assuming, on the epigenetic loss as.
Oh, we can talk about that too. You talk about well, that was the initial one that was referred to with the clock gene, correct?
I think so, but I haven't read the new paper by Sinclair. We can pull it up right now, though. But yeah, see if you can remember what was the other one that you wanted to talk about.
I'm trying to remember which one it was.
I do have a question, because you said you were getting to microdeletion as a gene mutations. What made you interested in the microdeletion side of things?
I mean, you know me, like I'm a SNP hunter, you know what I mean? Hunting all those polymorphisms. But so for all you guys listening, so SNPs, or single nucleotide polymorphisms, are when individual letters in your DNA are misspelled. So instead of a T, you have a G, or instead of a C, you have an A, something like that. Microdeletions are when portions of several, anywhere from tens to hundreds of these letters are deleted out of the DNA at any given time. Those are, self-decode does do some insertions and deletions, but it's mainly, SNP-based with probes. But the microdeletions just seem really, really interesting because they can explain a lot of mysterious, at least what I've seen the context of mysterious neurological type stuff. I think that's mainly just because, not because they're mainly happening only in neurogenes. It's just because that manifests much, much easier. If it happens in a gut gene, you might just have like bloating all the time, which is a lot harder to really pinpoint to something versus if someone doesn't have proper social cues, or it looks similar to stuff like on the autism spectrum, but it's not the normal criteria. And as technology is coming out, they're seeing that it's these microdeletions that can explain that. I just think it's just interesting how the infinitesimally tiny amount that we know about DNA now, how much we've learned about this. Even working with like we have a fraction of a fraction of a fraction of the information that we really know about, that we'll eventually know about the DNA in our lifetime or past that, and already been able to help people in so many ways. And I know you do some of that stuff too with Nebula, doing the whole exome sequencing. I think I'm actually going to get some of that too. Now, do they have do you just get a normal account, or is it like a pro account type thing, like self-decode?
No, they have the tools available for professionals, but it's a normal account. And if you know how to use it effectively, then that's pretty much it. There's a lot of bioinformatic tools that they provide too. It's a really good, I highly suggest doing just a genome analysis with them. Maybe they'll look at, for example, microdeletions, I'm sure. In fact, they look at mutations in introns and where certain alternative splicing sites may be just through bioinformatic analysis. It's so powerful if you really know what you're looking for.
Yeah, if you guys that don't know, so you have the whole exome sequencing is literally reading every single letter. Like 23andMe or self-decode, Ancestry DNA, they'll take probes and read large segments of the DNA. Now, so you have that, and then self-decode will take those probes, and these mutations happens in blocks on chromosomes. So they take the artificial intelligence and can infer, all right, you have this mutation, therefore this other area in the block will be mutated as well. So that's how they take, I think 23andMe is like 75,000 mutations and bring it up to 100 million just through that AI there. But then whole exome sequencing, which is what Nebula Genomics, which we were just talking about, that's literally from the get-go, reading every single letter in the DNA. That's honestly the future for sure. Like once you get that done, you're literally set forever. You know everything, every letter in your DNA. And then as science comes and slowly proves these things, what they do, you can go back to it. The reason why I didn't do that initially, it's more expensive right now. I think it's I think it's 700 or so just for the sequencing.
I think they had a discount. I'm not sure if it's still up now, where it's 600 for deep whole genome or whole exome sequencing from telomere to telomere. But then if they don't have the discounts, I remember the price is somewhere around like $2,000. So very expensive.
2,000. Oh, way more than 700 then. Okay.
Expensive. That was a discount.
Yeah. Okay. Yeah. So, in the future, as time goes on, that's why I want to get the Nebula and learn that, because that's the future for sure. And I don't know whether it's 5 years, 10 years from now, eventually that's going to be $100, just how, as technology goes. I mean, if you look at the pricing of these DNA kits, like 5-10 years ago, doing what 23andMe does or doing what self-decode does for $100, I think was like $85,000 or something like that. Like, it's crazy how it keeps going down.
Innovation gets better. Yeah,
absolutely. What are some of the with your clients? What have been some of the major intron deletion type things? Like, what type of stuff, like specific to Nebula Genomics, have you seen working with your clients that are
only one client has done Nebula Genomics right now, and she did it specifically because she has a host of unexplained issues relating to like postural orthostatic tachycardia syndrome. And there's no, and Ehlers-Danlos as well, the one that she has isn't like the really bad forms that you can pinpoint to a specific coding gene. The one that she has is unexplained at the moment by biology. There's no sets of genes even that they can figure out that are causative in it. But as soon as we did the genome test, and what came back were a lot of SNPs in intron regions, particularly for cardiovascular disease, collagenous tissue diseases. I forget which one was the one that was the highest risk, but she had high risk of coronary arterial dissection, which is when the endothelium itself just splits. It's one of the worst conditions that a woman can have. It's worse than having a heart attack because it fully causes a thrombus that can't be cleared without surgical procedure. And a lot of people die from this without even knowing it. It's one of the... Go ahead.
No, I was saying, when you said collagenous, were those ones that are typically implicated with Ehlers-Danlos, or they were like kind of ones that maybe have some data, or like it was like...
It depends on what the condition was, and there are some where you have thousands of SNPs that they map with very high significance values to like Mendelian randomized studies, as well as whole genome analysis, observational studies.
What's your email, by the way? I made the little thumbnail. I'm going to send that over to you.
My first name, Pedro, then the last name, Doamaral.
D-O-A-M-A-R. D-O-A. I should know this.
Doamaral.
My close friend.
Yeah.
D-O-A-M.
A-R-A-L. Doamaral.
At ProtonMail.
At protonmail.com.
Okay. Awesome. Well, now I'm trying to think. I wonder if this will work. If not, whatever. It's not the end of the world.
What is it?
Because, like, I don't know if you're going to close ad. It's weird. When you come out of the lives, you have to have a thumbnail ready to upload. But now, since you have to actually download it, I guess. But
we'll try it.
Yeah, we'll give it a shot.
If not, we got our faces. It works. It's the faces anyways. You're just making it into like a nice photo.
Yeah. Just so people know, what it is. Am I going blind here? Where is this?
I'm going to try to post this on YouTube as well because somebody had mentioned, and hopefully we can do it easily. In the meantime, I guess I could go over some questions. Let's see. Do you guys know anything about visual snow? Yeah, so visual snow itself, there's a lot of different causes, and the most common that's
visual snow. What is that?
It's basically where people see like little tiny speckles in their field of vision. It's not like a really bad pathology. It's just annoying, and it comes and goes for a lot of people. One of the things that can be a factor involved in this is fungal diseases, and it causes really bad black spots around the eye itself. Like as soon as you open your eyes, you see these black spots just traveling along with your vision for really bad fungal diseases. But from what I've gathered, it's a couple of things. It's damage to the retina itself. It can be damage to the collagenous tissue around the eye. It can be damage to the optic nerve. It can be damage really bad in the brain itself. It can be meningitis. It can be encephalitis from viral diseases. There's so many different causes. But in general, the most common one is breakdown of the collagenous tissue around the eye. And you see that as little speckles in your field of vision.
So this is internally, they're seeing black spots, not black stuff around the eyes that's externally visual.
The black spots are with fungal diseases in specific. Usually with visual snow, it's translucent or transparent. It's like you can see through it.
Different from eye floaters.
It's like eye floaters, but it affects almost your entire field of vision.
Interesting.
Some people experience it so bad that they say it's like static, like the static TV in a sense, and that's their field of vision.
Oh my God.
Purple spots though, so that's different. Yeah, like static TV, what I mentioned. And let's see what other questions we got. I just started med school in July. Any recommendations for pre-indoctrination reading? I actually don't think I've seen a lot of medical school curriculums because I was going to apply to med school, and they're really good. A lot of pharmacology, a lot of advanced anatomy and physiology, and within a very short time frame too. I don't think anything's bad about it, and I don't think people will be indoctrinated in any sense. I think you should take all of that information, but there are a lot of supporting agencies that fund medical schools that like to promote some of their stuff. Like the nutritional side of things is supported a lot by these big companies, Coca-Cola and so on, dairy and meat. Companies like Tyson, and you'll tend to get mixed intake depending on the medical school. And so those things you should take with a grain of salt. But more deep science like pharmacology and so on, you should absorb with everything that you can. Study really hard for that stuff, in my opinion.
Yeah, no, I definitely agree. And, I did the whole conventional medicine, pharmacology, PhD training and everything, and left that space to now be doing the more holistic stuff. But I wouldn't change my career path for the world. You know, like,
Hmm.
yeah, I had to learn how to do all these experiments with mice and isolate immune cells and stain them and this and that. All these skills I would spend months learning, knowing in my head that I'm never going to use this in 6 to 8 months. I would literally have to
That's so
learn
true.
from scratch because that in order for a PhD to graduate, you have to publish a paper that, you know, people haven't done before. And if you're the head researcher, you got to be the one doing that. Yeah, I had a couple of undergrads that would help me, you know, genotype my mice, like minor things like that. But the real hard stuff I have to figure out myself. And, you know, my boss hasn't done that for 20 years. She's just like, figure it out. You know what I mean? But I wouldn't change it for the world because having that deep science education,
Yeah.
point blank, you're not going to get education like that in the holistic space. You could take these courses to understand these things, you know, absolutely. But understanding the molecular biology, cell biology, pharmacology, neuroscience, like these conventional institutions, you know, they are the holy grail for that right now. But clearly there's a lot of issues with the context in which they're spoken to. You know what I mean?
Yeah.
That's why I'm so excited for what we're both doing, trying to merge the two there.
I agree. Let's see. Somebody mentioned self-decode shows one of the mutations that reduced MTHFR by 30%, and it came back under typical. Should I use folate, B12, etc., or should I adhere to other or to their recommendations?
Yeah, so good question. So hard to say, mainly because self-decode's great, but I do think I mentioned to them they need to with the methylation reports because methylation is much more than just MTHFR. Like for the money you're spending, I think it's like a buck 70, $170.99 a year for that value. It's no doubt in my mind worth it. But like, is it perfect? No, there's no. I think I decided to work with them because regarding the probe base. So the nebula seems like the king for, you know, the whole exome right now, but I want to do the probe base
Yeah, of
so it
course.
was accessible to people because people can't
Hmm.
pay 2,000. You know, I'd
Yeah.
rather do that end first than work up to that. But methylation is a lot more than just MTHFR. You have genes like the CBS gene, the BHMT gene, the MTHFD gene, NTR, MTRR. All these different genes interact in a whole cycle together. So if you have one thing with MTHFR that's messed up, it kind of depends what else you may be needing. But having one mutation with MTHFR, the two they report on, I think one, I forgot the RSID values, but one of them is more significant than the other. One of them lowers it by about 40% per mutation. So if you have one of those mutations, you'd be at roughly half efficacy of the MTHFR enzyme, in which case I would definitely consider trying supplementation. Now, however, I would also look to see if you have a COMT mutation. That's the gene that lets you break down dopamine and adrenaline. We mentioned that earlier. I think we dove into that in our prior live. You know, that's basically what lets people have higher dopamine, higher intelligence, alertness, but also higher mood swings, more prone to stress. On average, more extroverted type personality. But if you have that mutation, I do, and I tried methyl B vitamins due to my MTHFR mutation, I had really bad anxiety. And that's very common thing that I see. One of the first things I do when I start with clients is figure out what type of B12 vitamin is best for you. Do you have mutations in absorbing it, transporting it, activating it? So activating it would be the MTHFR mutation. But if you're not sure about the COMT mutation, you can try adenylyl and hydroxyl B12. Those are the two forms that I take. I noticed much cleaner feeling energy. I didn't feel sometimes shortness of breath. I don't feel very short with people, but with the methyl B12, I can't really tolerate that at any effective dose. I mean, the second I get even around 20% of the daily value, I just start getting these anxiety-like side
Mm.
effects. So, yeah, hope that
What what do you
answers.
what do you think that could be from in terms of the methyl B12? Is it just increased amounts of methyl groups interacting with certain enzymes, particularly, I'm assuming like the ones that produce
Yeah,
neurotransmitters, and is that what it is?
yeah, that's exactly it. So basically, you get more methyl B12 inside of your cells. You're making more SAMe. SAMe is used to make
Mm-hmm.
neurotransmitters, dopamine, adrenaline, and then people with the slow COMT mutation I break down adrenaline 4 times slower than the normal person.
Right.
So if I raise my adrenaline, I don't know, 20% from the methyl B vitamin,
It's
that
gonna be there for a
20%
while.
is I'm gonna keep making more. It's gonna
Yeah.
keep lingering there for longer, and it just keeps aggregating, building up more and more over time. And then I feel this, and we come down slower. So like when I get stressed, I've got a lot, the genes tell a lot, but proper supplementation, breath work, mindfulness, that helps a lot as well. I still break down adrenaline much slower than a normal person. However, I've done a lot of work to get better at containing that. So like genes can lock you into certain things, but not like it'll never change at all, if that makes sense.
Yeah. I'm interested in knowing exactly what the kinetics are. What you mentioned that you break it down 4 times slower with the specific genotype that you have. I would be like highly interested how they measure that stuff too.
Yeah, I mean, there were 2 or 3 different studies that were doing that. I think they were checking the level. I think it was mouse brain. Someone just asked a question about genetic tests are expensive if you're not in the U.S. The shipping for the self-decode kits, it goes from $12 if you're in the USA. I think it might be like $8 if you're in Germany. That's the lowest. And the highest it is $55. So with the kit, I usually order for my clients and then we work together. Or if you want to get self-decode account, it'd be an extra $55. So basically $154 for the DNA kit itself. I think with self-decode, you can't order just a DNA kit unless you're a practitioner. You have to also get the package, which is another $70, then $99 per year, which I think is more than worth it. But again, like someone mentioned, you can't cater something like self-decode to a mind like mine or yours with all of these nitty-gritty details. It's meant with the smiley faces, whether it's high, medium, low, red, green, yellow. Like it's supposed to be understood and to get actionable tips for people that don't know science. So you can't add all of these super deep, nitty-gritty, nuanced things into that. Then it turns into, a clinical grade platform, which it seems like Nebula is more on the sciencey clinical side, correct?
Yeah, it is without a doubt, especially more specifically bioinformatics, like a lot of good computational tools.
Yeah.
Somebody mentioned really quick, just want to answer. What B12 do you like instead of methyl, adenosine, or hydroxy cobalamin? Or
Yeah, I honestly
adenosine
like.
hydroxy
Yeah, I
B12.
honestly like both. The multivitamin. I take the pure genomics from pure encapsulations, and that's 50/50. And I feel really, really good on that. It's also got the bioactive B6, bioactive vitamin A, basically everything in forms that are ready to be used by the body, just to take a lot of these mutations into account. Yeah, those are the 2. So yeah, it's the cyano, which is the cheap one with the cyanide molecule. Then you have methyl, then you have adenosine, then you have hydroxyl. So those are the 4 major groups there. Adenosine can be really, really useful for people, especially with energy issues, because that's actually the form that is used in the mitochondria to make ATP. And then hydroxyl, I'm pretty sure it's hydroxyl. Hydroxyl can actually be turned into methyl B12 as well. So the thinking with that is you should always have some degree of methyl groups floating around in your body. So even though I don't take any methyl B12, there should be a little bit of methyl groups in there so I can methylate the hydroxy. And even though I do have MTHFR and don't do that as well, again, you'll never perfectly supplement around every single mutation you have. There'll always be something that seems not perfect because you got to go by how you feel at the end of the day. If a certain supplement may be better for me based on my genes, but like, for example, I have some mutations and glutathione production issues. And I take glycine. I love glycine. And acetylcysteine, I never really like how it makes me think, it doesn't make me feel horrible. I just don't really like how my thought process, how my cognition alters with it. So I honestly just don't take it. Could it maybe in the long term help my glutathione production? Yes. But I just go based off how I feel. So at the end of the day, always go based off how you feel. You have this information about, again, like I said, a tiny fraction of the genetic information that we're going to find out eventually. But for all you know, we could find out 5 years from now, you have another mutation that explains why you don't feel good on a certain mutation, on a certain supplement.
not N-acetylcysteine, sorry, CBS mutations that you had, right?
Yeah, I have CBS as well.
Yeah, it makes sense why it doesn't help you because N-acetylcysteine converts to cysteine and then converts to dihydrogen sulfide with CBS, and that's usually going to cause sulfur species to be formed, not in a favorable way. So glutathione skips all of those steps.
Yeah. Yeah, and then the CBS mutation, that's like, for example, that's another one that I pay a lot of attention to. One of the first genes I looked at with clients regarding methylation. Most people have overactive mutations. So you convert homocysteine into taurine, sulfur, and taurine, sulfur, and ammonia at the expense of glutathione. So there's a lot of different, instead of making glutathione from homocysteine, I'm making it into ammonia, taurine, and sulfur. That makes me very sensitive to sulfur. So like if I have a lot of onions or garlic, I'll get a histamine reaction because I'm very sensitive to that. I could have some, just not a whole crap ton.
Mm-hmm.
That can also make people sensitive to ammonia. I personally tolerate L-glutamine very, very well. I have a heterozygous mutation in one area on that gene. So it's relatively minor, but the sulfur still bothers me because I also have histamine-related mutations. So it'll kind of like piggyback off of that. But I've seen clients that have, homozygous mutations there or double mutations there. And I always try L-glutamine. It's perhaps one of my favorite gut health subs. Helps seal the leaky gut, helps with hyperreactivity and food allergies, food sensitivities. But it also can be converted into ammonia. So people that have those mutations, if you feel bad when you take L-glutamine, odds are you have this mutation and you're just very sensitive to ammonia. And you could take L-ornithine to lower the ammonia that you're taking from the L-glutamine, but at that point, it's kind of like, is it really worth? It reminds me of the doctors saying, take this medication for the side effect from your other medication. So, obviously, they're natural things, but still, I'd rather just try no glutamine, do something else.
So you're specifically saying so people are aware of symptoms, neurological symptoms, right?
Yeah. Sometimes nausea, but it's usually fatigue, nausea, confusion. Like, I wouldn't call it like you don't know what the fuck is going on, but just kind of like the cognition feels very blurry and you just feel off. And that just means, again, you're sensitive to the ammonia from the L-glutamine. But even if people do have that mutation, I think the smallest bag from bulk supplements is like $9. I always tell them, listen, you have these mutations, pay attention to how you feel. I think it's worth risking the $9 because if you can tolerate L-glutamine, it's one of my favorite supplements. I take that every single morning.
You mentioned earlier in the conversation about the roles of exosomes with specifically how the cells are transporting things around. And we talked about microdeletions and coding regions. I want to combine the two and mention something I think might be interesting to you. What about possibly deletions, frameshift mutations, any kind of SNPs in non-coding regions that also are regulated by RNA transport through exosomes? This is something I got interested in quite literally yesterday, and I'll explain why. There was a couple of papers that were showing resveratrol, and taking resveratrol causes a shift of fast twitch muscle fibers into slow twitch muscle fibers. And some researchers went in depth further to categorize how exactly this is happening. And it seems that resveratrol causes an increase in a type of microRNA that then interacts with non-coding regions to promote this switch from fast twitch to slow twitch.
So it's like lineage differentiation. Like they're literally changing. Are they behaving like different muscle fiber types?
Yes,
or they're literally like convert? Is the behavior different, or they're actually changing permanently?
If you go from fast twitch to slow twitch, a lot of things have to change about how cells are metabolizing fuel sources, especially. So in that case, it made me think about, once again, the non-coded regions being really important because they interact with so many different forms of RNA that are produced. What if you have, for example, an SNP or microdeletion in a non-coded region that then prevents the proper binding of RNA or enzymes that utilize RNA to elicit effects afterwards? That is something that I think is so underexplored, but so important and probably could explain a lot of the things that seem to be more environmental in nature. So, you know what I mean?
Well, so guys, microRNAs are basically like, imagine like a silencer on a pistol. MicroRNAs literally bind to an area and will prevent it from being expressed. So when he mentions environmental, because environment are things that change. Like when people say I felt great my whole life and now I feel horrible, genes can help explain that, but that's a lot kind of what he's mentioning there, like a shift due to the environment. And regarding those exosomes, that's one of the main things. They deliver proteins, regular RNA, and microRNA, even peewee RNA. But the microRNAs are the main thing that's studied because it can change the behavior of these things, of those. That's super, super interesting. And I wonder if any, I feel like that's got to be strictly in the academic space with the R&D with that. I don't really know of anyone that would be looking at the mutational landscape of the, because it's always the non-coding region. So meaning, to all you guys, you have so you have areas of a gene that make the protein. So we mentioned the COMT gene. That's the coding region that makes the protein that can be mutated so it doesn't work as well. You have the non-coding area, it never gets made into a protein. So that more so regulates what gets made, what doesn't, or you can make a version of a protein that's truncated or shorter. So it may have different processes inside of your cells. That is really, really interesting. And I wonder, again, how many years ahead of this we're thinking about when we'll actually be doing that. I mean, people do that type of stuff. It's pretty similar to, I did some microRNA exosome work in the context of cancer, breast cancer. So how does one cancer cell convince other regular cells to become cancerous? They'll deliver them microRNAs. Dude, exosomes are so fucking cool. You can have breast cancer cells deliver microRNAs from the breast through the bloodstream. These little exosomes will float around. They'll go all the way to the lung. And the microRNAs will change the behavior of the lung cells to make them inflammatory, to recruit immune cells, to basically cause inflammation, like the pre-metastatic niche. So basically, before cancer cells metastasize, they'll make the area they want to go to more habitable. They'll make it inflammatory, which cancer loves. The fact that a cancer cell in the breast can like send this package across the body and change. I like to think of it like you're auto starting your car in the morning on a cold morning. You press auto start, so it's nice and warm when you get in the car. They're pressing auto start and making your lungs inflamed. So by the time they get to your lungs, the actual cancer cells, they can set up shop and grow. But yeah, regarding the microRNA stuff, that's so interesting. I think that could describe a lot of different things, especially regarding things maybe with heavy metals or, mold. Because I honestly think like, how many people do you think that get heavy metals or get parasites or get gut infections or get mold toxicity? How many of them do you think actually have mutations associated with these pathways? Because you have the susceptibility of the individual, so the genetics of each person, then you also have the load of these toxins in the world. So if there's high enough levels of metals or mold, even if you're not mutated, I think you'll eventually get infected and get sick from it. But I would think that most people that get these things have to have some sort of mutation, especially if you're living in the same house, in the same vicinity as other people, and you're the only one getting sick. That's clearly genetics. But just in general.
There are actually, I think you're spot on. I do remember, for example, specifically with mold toxicity and the mycotoxins that it produces, more specifically aflatoxin, it intercalates in DNA. And so it sets shop right in between DNA. And so the strands can't be read anymore. It's just a purely modified strand that needs to be removed. Though the mutations that are involved in how effectively this gets removed are with polymerases. And if people have defective polymerases, they won't heal as quickly because your DNA isn't being read in the first place. It's completely destroyed. So no transcription factors can be produced. No protein-coded regions are going to be read in the first place. It's just a pure carcinogenic form of DNA that fully depends on excision or endonucleases in this case to remove that area and then build upon it once again to make it fresh. And so in my opinion, it has to do with a lot of DNA repair processes. BPA is another one that sets itself right between the strands of DNA. Other phenol-like compounds do the same thing. And aromatic hydrocarbons, of course, do it as well. So air pollution is a big one in contributing to a lot of these disease pathologies. And the only way that it can be fixed is through removal of that region that's been affected by the pollutants in DNA.
So is it mainly DNA polymerases or the endonucleases?
Both.
Like which?
Both.
Okay, interesting. So those probably would, yes, that would be directly linked to cancers as well. Just because anything that's causing those DNA repairs, I think Was it your presentation that you had? I think that you shared a slide on Instagram with the elephants and the polymerases, or the blue whales and polymerases or something.
Yeah, that was a while back in summer. Yeah, blue whales in specific, they seem to have not only higher concentration of tumor suppressor proteins, specific forms, they also have increased endonuclease and repair, base repair enzymes.
Yeah, so for all you guys, it's just like that scientists wanted to look into that because you have these animals, mammals that are so much, so their cells are, they're mammalian cells, so they're relatively similar to ours. And they have so many more, their cells are bigger too, I think, right? But it's also, it's not just the cell size, right?
I'm not quite sure.
Okay,
I would
well,
assume they have to be bigger.
yeah, yeah, I think they're a little bit bigger, but they're also so many more cells to make up an elephant or a blue whale. And why do they not get cancer? Probability wise, all these incredibly higher number of cell divisions have to occur. You would think there'd be a higher rate of error. But as he was mentioning,
The repair process.
the repair process, they can repair their DNA so much more efficiently. If we can figure out how to repair DNA better in humans, that's going to be the main driver for cancers, because I think I think it's more so the DNA damage is what initially, my head's going to explode.
That's funny.
The DNA damage is what initiates the cancer. Now, inflammation on its own can cause DNA damage, correct? Even if it's just TNF alpha interleukin 6. It doesn't have to be BPA or something else. You could just have high inflammation from leaky gut from bacteria. So not necessarily, I guess bacteria is considered a pollutant, I guess, but you get what I'm saying. Like it's not like a heavy metal or a mold. It's just like
they still interact with your own DNA, mRNA, and I do believe there has to be some other forms of RNA, and bacteria do communicate with our own DNA and exosomes and microRNAs. There's communication between all kinds of organisms, and that in turn will affect the way that DNA is transcribed in the first place. And so it can completely, somebody can be totally healthy, no mutations, but the interaction can push it in a manner that reduces expression of a certain gene that makes it seem like the person has an issue with that gene. There's so many interplays in general with like DNA that isn't considered and only seen mostly in the literature. You don't hear about this in mainstream communities in functional medicine, Western medicine in general. It's mostly just researchers analyzing this and seeing that, wow, what we thought was this just one coding kind of concept is actually a really flexible system that can be interplay with many different organisms, pollutants, compounds, etc. It's like DNA itself is not a static thing. It's the most dynamic thing that you can think about in your cells.
Yeah, and then the whole what is it, synthetic life type stuff where they're just actually programming they're feeding. I mean, you could see it. You probably know more about it than me. They're basically just feeding are they artificial cells, or are they like anucleated cells? Like, what do they actually put? They put DNA into something that literally makes artificial life, that actually makes these proteins.
It was Craig Venter, and from what I remember, he took a E. coli bacteria and basically removed all of the DNA from it. So it only had the cytosol, and there's no activity, it's just a cytosol, basically like a big liposome. Mm-hmm. And then he synthesized fresh DNA through chemistry. It's pretty simple to do. You make the nucleotides and link them together. And with it, he added it back into the cytosol that's completely devoid of any kind of genome, and made a fully synthetic organism that functioned just like it was supposed to do. It's insane.
Did he replace the DNA with the same DNA to see if it would work? No. Or did he tweak anything?
Not that I'm aware of, but he took fully synthetic. He made it in the lab through chemistry.
But I'm saying it was the same as the DNA he took out, just to see if
I think he made a completely new one. He made it with specific genes that he wanted.
Okay.
Which is crazy to
think about. Yeah. You could be naked. I mean, I feel like that's how you could be. I mean, it reminds me of like the yeast, how they like, pump out pharmaceuticals and stuff through yeast, except this could actually be, you can only control the yeast biology so much. If you guys that aren't aware, like, for example, I know that for psilocybin production, so now that psilocybin's crushing these clinical trials for pharmaceutical use, they're not going to be using raw ground up mushrooms in the clinic. It's just far too variable. Like, you don't know, even if they grew them in the lab, you need to have the exact same doses of psilocybin So they're looking for ways to produce psilocybin at scale. And the amount of psilocybin mushrooms they would need, it would just, the cheapest way to do that, you can actually put these genes to produce these things inside of yeast. And you could grow yeast much more in a much lower space, much lower cost than growing actual mushrooms. That's what it reminds me of. You're giving the genes to the yeast, but even with the yeast, you can only alter their behavior. You can tell them what to make, but like you can't really alter the behavior from scratch. Because at the end of the day, the difference here is the yeast DNA is still there. You're just giving it a couple things. Hey, make this, make that for me. This is literally from scratch. You could just be generating it.
Yep. Purely synthetic biology. It's very interesting stuff. I definitely want to get into it for my master's, specifically with like chemistry.
What don't you want to get
That's
into?
True, man.
I think that's it. What scientific topics are you just like, no, not for me?
Political science. All right, let me keep going. We got some other questions. Do you agree with the blood type diet? What's your opinion on that?
I haven't looked into it that much. I do think, I'm not sure if it's specifically just the blood type itself, but I definitely think there could be genes that dictate blood type that also may be affecting diet, if that makes sense. But I haven't done that much reading on it. But I think that I haven't done that much reading on it. But at surface value, I think that you can tell exactly what foods agree or disagree just based on that one variable. Seems a little far-fetched to me.
You know, I used to read those blood type diets back before I even knew what exactly constituted blood type with the sugars, what is it actually? Polysaccharides that stick out of cells? Even I should know this by now. O type, O is basically no, right?
Yeah.
So I used to look at those charts and it would say that O types should eat more meat and they should eat less vegetables. B types should have a vegetarian diet and so on. So they had these different these differentiators based off of diet for blood types. And I've always experienced a positive effect following that, even if it's completely wrong. So it might be true in certain contexts, maybe with people that have other things that affect diet composition in a favorable way that align with the blood type diet. But I do know from COVID research, there are differences in people that get really bad infection, specifically based on blood type. So there has to be some interplay here that does actually, it exists in a sense.
Yeah.
Like,
well, they've known that even pre-COVID. Like everyone, obviously with COVID, they got a huge resurgence. But that literature about regulating viral infection, that's been out for, I want to say at least 5-10 years. They've known that, and I just feel like if the blood type diet were universally true as gospel, then blood type should be very based on specific evolutionarily populations. They should be very, everyone in certain areas, evolution, a lot of stuff. The more I get into this, the more evolutionary biology seems cool. I used to love dinosaurs and stuff, but then evolutionary biology, in undergrad and stuff, you learn the phylogeny trees, and I'm just like, that's not cool. It's just like, I don't really care what sea sponge came before which mollusk or whatever the hell it was, But at the end of the day, like, what diet's best for you? It depends on where your genes are from. I know you made some posts about this with like the fruit and MTHFR and like what food was available to your ancestors. And going back,
we're getting into Paul Saladino territory. I don't know if it's a good idea. We might get attacked.
Bring it on.
Speaking of that, consider your mutations with CBS, COMT, MTHFR, and other ones that you've mentioned that you've experienced negative side effects from if you follow, for example, a specific protocol. If we look at it from an evolutionary perspective, why do you think that is, that with your history and your race and how far back you can trace your lineage, you have these mutations? You think it's just purely de novo neutral theory of evolution, where it's like you're bound to get mutations anyways? Or do you think there has to be some interplay here where something's promoting these specific types of mutations surrounding methylation patterns?
Selecting it, you're saying? Like
yes, yes.
I definitely think it's a mix. Honestly, like that's a whole new, I think you're definitely more well-versed on the actual evolutionary side of the genetic side of that, but I would definitely think it would be a mix. And I think one thing a lot of people that I disagree with, some people say, is that like, as if nature optimized our DNA now, like meaning that like, I'm trying to think how to word this, like just because like cavemen got multiple sclerosis, cavemen were anxious, like our DNA wasn't perfect back then. It's not perfect now. I think that you don't necessarily, there can be mutations that are a certain level of disadvantageous. Is that the right way to word it?
Yeah, works for me.
That would still permit survival. So you can have like a co-expression of things that may help. And then again, these mutations happen in blocks on chromosomes. So you could have one mutation occur that guarantees another mutation occurs. So they can kind of, you can have one that's so good that it carries the weight of one that's less than favorable. But I also definitely think too, mutations in like the FAD enzymes and how you metabolize and use like omega-3s and 6s, this and that. If you're having more, if you're at a evolutionary standpoint, more fatty fish, that could be beneficial to you as well. Like I said,
Right.
you made the fruit reference with the MTHFR. So I definitely think it's a mix, but I also think people also act like I said, our genes are at the final end state now, or even I guess pre-industrial era, because even that's still 100 years ago, 150, that's still a blip evolutionarily. And back then we were still pretty much, now in the modern world, it's becoming more and more of a genetic melting pot. So I think that's also why, like in my practice, I see people very high prevalence of like multiple homozygous mutations or compounding mutations and stuff. And I'm like, is it really that like this more than half of people in the world have these mutations? And I think part of it's because, a story rant about this a day or two ago. I think people that search out people like us, if you're searching for and find a holistic genetic health optimizing coach, clearly you've tried all these other things. And I definitely think that you tried all these other things, and I think that that's partly because you have less than favorable genetics. Again, because you're breeding these populations. Even if it's still like I'm thoroughbred like Caucasian European, but I also have different areas. I have some Scandinavian, and I have some Irish. I have some, so it's like still different subpopulations that did have these different diets there. So overall, I definitely think it's a mix as usual.
Yeah, that makes a lot of sense. No, I would completely agree with that. I think that there's definitely some selective factors depending on what it is, and then there's the de novo factors that you get a mutation. Even
you want to explain that for them real quick. The de novo.
De novo meaning even now, if you're outside, for example, for a long period of time and you get sunburned, what's happening to your cells is there's DNA damage that is occurring. And if the DNA damage occurs in a cell line, that can then differentiate or proliferate and then differentiate into other cells, meaning like one skin cells or one skin stem cell in specific becomes a different type of skin cell, but it carried a mutation from the beginning when you were sunburned. Well, then you have de novo mutations, and that can be carried out as well through sperm cells, egg cells, if you have damage to them. And so you can have babies that are born with mutations de novo, meaning newly acquired mutations that weren't there before.
Yeah. And just to add a little clarity there, like the skin mutations would not be, it would be the gamete, the sperm and egg mutations that get passed on.
Exactly.
Correct. Like the change in DNA on the skin, that would be affecting me. Let's say I got something that would be affecting me, but that's not in my sperm cells. So that will not be passed on to future generations. And that skin cell can't like travel to my gonads and then, turn into like that wouldn't really occur. But, and I think that's too, when they were doing those CRISPR twins, it caused a lot of upset in people a couple of years ago. In China, I think they were doing the CRISPR twins. And the main issue was, like for CRISPR, for like, I think like muscular dystrophy, they will do gene editing on muscle cells. And those muscle cells, people are more okay with that. Versus with those two CRISPR twins, like two little girls, like in utero, whatever, actually no, preconception, they did in the sperm cells and edited them and then fertilized. They did a thing, an HIV gene.
So it was the chemokine receptor for HIV. I'm pretty sure I actually just read the book with the whole whatever happened, and the Chinese scientists did it without the government knowing, and essentially he got backlash for it, and I think he went to jail.
I thought he was in hiding. Did they find him?
Oh, I'm sure they did. As a scientist, you're not going to get that far.
Yeah, but I thought it was so crazy because they deleted that chemokine, and then they were resistant to HIV, and then they say they're age 2 or 3. They're like, oh, what do you know? These children are also hyper smart. the genetic editing also made them very smart. I started laughing. The first thing I thought was, you're telling me China didn't, these top genetic scientists didn't research what else it does in the brain.
Of course.
They knew.
Of course.
Their goal was not to prevent HIV.
Yeah.
Their goal is to create genetically, intellectually superior, genetically smarter humans to take over the global scale. And we're going to pitch it as, you know, the Trojan horse of it so happens to do HIV. And that goes to show, guys, you can have one gene affect many different processes in a cell. You can have a mutation. So, for example, this protein that affects chemokines, that affects HIV infection, also clearly affects neurotransmission. That's how they became smarter. But the real issue people had with this was, I don't know how old they are now. We're not going to get any updates. They've got to be at least 5 or 6 years old now. That mutation from the CRISPR, the editing, is in every single one of their cells, including their egg cells. So the problem now is those mutations can now be passed on. So we don't know down the road, you mix that one mutation. Remember the Punnett squares, guys, you know, the big A, little a stuff. If you pass that on with someone else with a certain mutation, you may be either messing people up more, meaning like making detrimental to health, or it could be messing them up, meaning making them smarter, more resistant to infection. But that's the real fear for this designer babies. You know, you can pick what genes you want your baby to have, because then we're introducing new genes into the gene pool. And again, as we're slowly becoming a melting pot, all these different continents, countries of people are slowly interbreeding together. You don't know what mutations could be introduced. Then you may get, you know, 20, 40 years down the road, just a bunch of embryonic lethal, just huge miscarriage issues, you know, because you don't know what's going to happen down the road and could cause depopulation or something like that.
Bro, speaking of Punnett squares, I remember in genetics, I think it was like an 8x8 Punnett square. Imagine how much information you would have to acquire if we didn't have bioinformatics, for example, through Punnett squares for every possible gene interaction. It would be never ending.
Yeah. I mean, even self-decode, some of their polygenic risk scores, I've seen over 1 million genes into account. I forgot which report it was. Might have something like joint pain or inflammation, but like they have 1 million different genes that are associated with that, and they follow that interactome, you know, and it's like 1 million different genes that some of the reports, you know, they'll only have like 10 or so, you know, because it's just relatively preliminary. They go based on where the data is available. But 1 million different interacting genes that you said can make proteins that all clash with each other within 15 seconds in a cell. I'm just imagining literally the most chaotic, if you look at a cell in a microscope, you just see like stuff like this. But now I'm just imagining you zoom in on a molecular level, which most microscopes can't do. It's got to be a complete clusterfuck of just like, I'm imagining chaos, like high velocity, like not a slow transport. It's like the Autobahn everywhere in that place. I love this. Like, that's crazy.
I love the sound effects, Tyler. Oh, man. Have you ever seen molecular dynamics simulations of proteins and enzymes and how they're always like in a vibratory state? That's basically what's happening. And they slow it down. I believe it's to like nanosecond range. So it's still vibrating like that within nanoseconds. Imagine within a full second.
Imagine a time lapse.
It's insane. I think somebody.
So someone asked a question: are you not recommending self-decode? No, I recommend self-decode to anyone that's interested in learning more about their genes. My point is that I think that they're way ahead of the game with their artificial intelligence, how they interpret more genes out of your DNA, and how they make these reports. Like, I think it is rock solid, eerily accurate with these polygenic risk score reports that it shows you. You'll be like, this explains me, this explains me, this explains me. My point is that I'm very, very, very detailed and hyper nuanced with this stuff. So the recommendations are great. People do make these recommendations and improve upon their lives. My point is, as you really peel back the layers of this, you can have one recommendation that is right for a certain gene, but then may not be good for another gene. And that's not because there's actual data out there. That's because there's someone like me that's understanding the cell biology. So if you want to get a basic good look on your health and your genetics and your biomarkers, what to tell your doctor to look out for, like, I think it's a no-brainer to get. But my point is, for true optimization, I'm very, very in-depth and like neurotic about this, and want to go deep, deep, deep with everything. Make it totally perfect for everybody, each individual. that requires a lot more nuanced thinking. And that's where you will need someone like me to really break that down for you. But that being said, again, I think it's $170 for the DNA kit. A DNA kit and a full year of access to all the self-decode reports. So as they add more reports, you're going to keep access to that for a whole year, and then it's $99 a year after that. I definitely think that's a great buy. It just depends on the person you are. Some people, they just want to be like, hey, tell me what I need to start doing, explain it to me nicely, that's it. I don't want to go into these reports and keep looking at it. And if that's the case, that's why with a lot of my clients, I order the kit for you. You don't actually have to get the self-decode membership account, but you can always take that raw data and re-upload it after the fact if you want. So I'm not telling people not to do self-decode. It just depends on what you want. I've had some people that say, I got it done and it's a little over my head, it's complex. It just depends on what education level you're at. If you're like a health coach or have some science, even like undergrad science, like you'll get a lot more out of it than someone that, didn't go to college. Even though it's written at a 8th grade reading level, it's just the concepts behind it can be a lot to implement properly and not have any clashing supplements.
Mm-hmm. Question for you regarding what you mentioned just now about perfectionism with making sure.
Real quick, sorry, this other guy asked another thing. So the DNA testing, I use self-decode to do the DNA testing. I also use their platform as a framework for my analysis. So I use their DNA kits. Those are the kits that I order for clients. And I also use their reports. So the same reports you'll see, I use to guide my process. I just add further detail, further nuance to make it really hyper-personalized and make sure the protocols that are put together use the most effective supplements in a way that won't bother any other genes in your body, and also trying to use as little supplements as possible that address multiple mutations.
So in regards to exactly what you're explaining to him with the nuance and hyper specificity to help somebody just feel good, there's a lot of people that I'm sure you've seen some Instagram videos saying that the personalized medicine side of things with specific genome based therapies are not really producing successful results that you would expect it to produce empirically. What do you have to say about those kinds of studies in specific and empirical evidence in the face of all of the interplays that can never be fully manipulated in a clinical study with genomics?
Yes, I think this is a really, really good point. Something I plan on being a lot more vocal about is, I'm trying to think of an example, I read studies about vitamin D mutations and how, they'll check 2 or 3 different mutations, and they will, on 2 or 3 different mutations on 3 different genes, let's just say, and see how they affect vitamin D levels. And then they'll see how they affect infection rates, something like that, to show that the mutation not only lowers vitamin D, but it also affects infection rates. And the problem is, let's say they show a gene doesn't have any correlation with that. The problem is that everyone has possibly multiple mutations on multiple different genes. So it's that, again, kind of going to that polygenic risk scoring where you take massive amounts of genes into effect because they're all interacting together. You see no change. If I look at a study and I see X supplement isn't good for anxiety, let's say, if a lot of these individuals may not have a mutation that affects that pathway. Let me try to think, for example, let's say, I don't know, taking lemon balm extract that raises GABA for anxiety. There is data that it helps. But people that have mutations in GABA pathways will respond extra well to that because they're going to have lower GABA to begin with. So they're all the way down here. They can be brought up a lot versus someone with less mutations or no mutations. There's not going to be much of an issue there. So they can't really get that much more calm without going over and getting side effects. So I think the problem with a lot of these studies is you can have, let's say you have 100 people in the study and there's no difference. The dots are all over the place. You can have the top 30 or so people that have the best improvements. They may have mutations that actually match how that supplement or how that drug works. And then the people that don't respond well to it don't have those mutations. So at the end of the day, you're going to be washing out all the significance here. It's going to look like no change, but why are we treating every individual the same? So I don't really, I use science to guide. I use science to guide me how cells work, how they behave. Because at the end of the day, all of our cells behave the same way. There just might be more or less signaling in certain pathways. But regarding the clinical studies, what works for certain issues, I take that with a huge, huge grain of salt because, again, you don't know their mutations. If I see someone with, let's say, or we could say endocannabinoid mutation, and they don't produce as much of them, they will respond better to CBD because it raises endocannabinoids. If someone already has normal, or melatonin is an easier example for people. If someone doesn't have mutations in that clock gene that we mentioned, or melatonin, they produce normal amounts of melatonin, but they can't sleep. Taking more melatonin probably won't work that well for them because their body already has enough melatonin. They may have another pathway that's mutated.
Yep. This is comes down to statistics, really. The population you choose, they can have selection bias, for example, where you have people that really are affected by certain supplements with GABA, for example, and they will respond favorably. You'll have confirmation bias as well. But nonetheless, taking a bird's eye view of this, if people get improvements, what does it matter? Yeah, that's it. That's how I see things. Like, in my opinion, empirical evidence shows no significance for 30 people, 30 dots on a XY graph show no benefit, and then 15 dots show some benefit.
Yeah.
If we take the average, it's basically null. But what about those 15 people that
that's a really, really good point. I'm stealing that for sure.
Like that,
I'll cite you on that.
That's why I love math, man.
Yeah, that's really, really interesting.
Let me answer somebody's question. I think somebody had a really big concern. So let me go back to the question box. Do you guys know about reactive oxygen species injury from high dose vitamin C's causing pins and needles? How to reverse? Actually, yes, it basically just got to wait it out, support nerve regeneration.
Why does that occur?
I would think it has to do with glucose metabolism, since vitamin C still interacts with similar pathways and basically causing peripheral neuropathy. So I think you just have to wait, helping the nerves
interesting.
Heal.
So it's sort of mimicking, in a way, the diabetic
high glucose
neuropathy.
Level.
Because I think, vitamin C is absorbed. One of the two pathways it's absorbed in cells is through those glucose transporters. So that makes a lot of sense. Wow, that's actually so interesting. If you have so much ascorbic acid floating around your blood and it keeps going through the glucose transporters, you're basically effectively blocking them. So glucose raises. Wow, but when she asked that question, I said, I have no fucking idea what that's about. Yeah. And then now I'm like, that's so interesting.
That's just more physiology to remember among the mountain of physiology that exists. So yeah, in regards to this, how to reverse it, you're going to have to give it time. Your nerves are going to have to heal, and stop supplementing vitamin C for now. And then this other question was best way to detox gadolinium from MRI contrast. And it's actually something I've looked into for a couple of clients, and there's really no way, because the metal will come out based out of its own interaction with your body. The radiation that might occur, you can't.
What substance is
gadolinium, the metal.
I wonder if it shares any. Because I'm looking more into heavy metals. I wonder, gadolinium. Yeah, I wonder if that shares any detox protein pathways, with some other types of heavy metals. This is definitely more of a you question. Gadolinium, what metal would that be most similar to molecularly?
Let me see. I've got to go periodic table.
So should we not take ascorbic acid? It just depends on the person and the dosage and the duration. My multivitamin had, I think, 180 milligrams of ascorbic acid every day. People have gotten a lot of good results mega dosing for certain applications. There's really no universally good or bad supplement out there. And I really think anyone that outright demonizes or outright promotes for everyone any supplement, I think is just a clown. More to the demonized side. I think certain things like magnesium, universally good for most people. You just got to be aware, if you're eating a bunch of foods that are very high in magnesium, then maybe you may not need that. That being said, most people are deficient in magnesium. So anyone that says that there's universally a bad thing is just trying to either scare you out of taking something so you buy their thing, or trying to say it's universally good so you go and buy their thing. And that's kind of why, I used to think I want to have a supplement company. I don't know if I'm going to have that in the near mid future, just because I would want to make supplements that are hyper personalized to each individual. With my clients, I give them products, individual products that are pre-made that match their given thing. But regarding making them from scratch for each person, each custom blend ingredient by ingredient, I feel like I'd have to have 15 different SKUs, a bunch of different formulations, which would make horrible sense business-wise. You barely would be making any money because you have to have you don't just have one blend you make and give to people. So yeah, no universally good or bad vitamin supplement drug. They're just inert molecules that interact with your body, and based on your genetics and your environment, they can interact with your body more favorably or less favorably.
Yeah. I'm looking over here in the gadolinium stuff. Renal metabolism is how it gets rid. So if somebody has renal failure, they don't recommend it. In my opinion, there's not much that you can do except supporting with chelation. And it depends on what, how exactly they're using it for, if it's MRI or for some other scanning methodology. These heavy metals, it's tricky, in my opinion, especially with, specifically gadolinium is a metalloid. So it's not even like a pure metal. It's more like aluminum than anything else. It's tricky, in my opinion. Let's see. Let's go to the next question. What's your thoughts on NMN, nicotinamide mononucleotide?
You want to take it first?
I'll let you take this one.
Sure. I mean, I personally never supplemented with NMN. I think the data is very, very promising. I think Sinclair is doing really, really great work with that. But I also do think that sometimes the claims are a bit overreaching with longevity research. Like I think depression research carries over much better between different organisms. So I think mouse studies can be translated to humans much more reliably than something like longevity, like in between different types of species of animals. So it's very, very promising, but again, too, I do think that, for example, like NAC is really used for longevity, but NAC can also affect mercury binding inside of your cells. So taking a bunch of NAC unnecessarily and boosting your levels up could possibly affect mercury toxicity risk. So again, that dotted line that analogy I gave you guys, you want to find out what things are either genetically suppressed or through your environment it alters. Anything at the end of the day, it's going to be modulated by the genes, whether it's the genetic code itself hardwired or the environment affecting the expression of your genes. What's below the dotted line on where your cells should be? And conversely, what's higher than it should be? If you have mutations where you have too high of adrenaline, you can eat things that have less amounts of tyrosine, so you have less precursors to make the adrenaline. What was the question? Oh yeah, the NMN. Yeah, kind of got off track there. But honestly, I'm not a hardcore longevity guy. Not that I wouldn't say it's one of my top areas of expertise. I personally haven't used it, but I have seen good data on it in mouse studies. I'm not aware of any human studies on that yet. Are you aware?
Of NMN? Yeah, there's a couple. It's not like crazy evidence. I've seen better evidence with niacin than NMN. And
big floosh guy.
The floosh. And it, in my opinion, it's definitely useful for people that have, or that people that need to skip all of those tryptophan metabolism steps or the niacin metabolism steps and go straight to making NAD+. So it's useful in that manner. But in my opinion, I've seen better effects with niacinamide than with NMN.
And now, by you've seen, do you mean like anecdotally you feel better, or you read data? So you're talking about actual longevity, life extension stuff?
Not even longevity stuff. People with muscular dystrophy, with the genes that make you have muscular dystrophy and lower mitochondrial yields inside of their cells, when they supplement with high doses of niacin, within a couple of weeks' time, I think it was 8 participants, but 8 participants with muscular dystrophy is kind of hard to find more because it's a very rare disease. And when they did this, they supplemented, not only were their yields of mitochondria inside of cells much, much higher, so they had greater mitochondrial biogenesis, their muscular strength came back. They were basically totally normal once again.
Floosh or non-floosh?
Floosh method. So if you can imagine a condition with like muscular dystrophy improving with niacin, I think it's way better in my opinion than niacinamide. NMN. I want to just make one correction here. I mentioned gadolinium is a metalloid, and the reason why I mentioned that was because I was looking at gallium instead. It is not. So I was wrong about that.
Gotcha.
Periodic table of elements, metals, man. Too many of them.
Someone asked, is there something you recommend for a person with type 2 diabetes just starting lifting weights? So good job lifting weights. Lifting weights is great. And I think before we get into the supplement stuff, there's some data showing doing a 5 to 10 minute walk after each meal is much better for overall blood glucose, therefore reversing type 2 diabetes, than doing one 30-minute walk. In the summer, you guys saw my old reels. I'm walking around getting my daily sun exposure, just recording my reels. I would do that for 10, 15 minutes after each meal throughout the day to help regulate the blood sugar regulation. Chromium deficiencies are more common than you think, like genetic mutations with the absorption, and chromium is important for blood sugar regulation. But regarding supplements that actually have, I guess, anti-hypoglycemic effects, so they help lower blood sugar, increase insulin sensitivity. A lot of them that are used, like cinnamon bark extract, fenugreek, like those two, for example, are two ones that are commonly used. But be careful; those can be very useful, but be careful because they also block the enzymes that break down adrenaline. So, like me personally, I'm very sensitive to that. So I would take these, like I took, and berberine as well is very, very common. Very, very good data, possibly as effective as metformin, but safer. Those could be good things to try. However, just be aware that they may also affect your mood. Like I took dihydroberberine. So it's berberine that's modified to get absorbed much, much greater. I took that in the morning with my breakfast, my normal amount of caffeine, which really isn't that much, maybe 200 milligrams nowadays, first thing in the morning. And I was tweaking all day, like super anxious, couldn't sleep that night because I'm very, very sensitive to that. So those are some good ones. I really like bitter melon extract as well. I really have not noticed, I haven't found much data if it affects adrenaline, but anecdotally, I really don't feel that as much. So usually with clients, I like recommending bitter melon extract and have that with your meals, possibly in between meals. But you want to be careful. If you have too much of that and too little of carbohydrates, if your blood sugar is not high enough to handle that dose, you can go hypoglycemic and feel really, really shitty. I've done that a couple of times before I was figuring out the right dosage. I have some rice, but I have too much bitter melon extract, and my blood sugar tanks. I feel horrible, and I got to chug 2 Gatorades just to feel normal again. That being said, it wouldn't be like this is not like a life-threatening hypoglycemic thing. Like if you did too much insulin, for example, that can literally kill you if you inject too much insulin with not enough carbohydrates. This would be more so you just feel kind of That's just for some context for you so you're aware on what to feel. Because one thing I'm very vocal about, I need to make more content about, I see a lot of people taking the wrong supplements that are making their issues worse, whether it's inflammation worse, making their depression worse, making their anxiety worse, making their energy worse, because supplements can interact differently with different people.
Good points. I like your mention in the beginning about walking. It's basically GLP agonist, essentially, a lifestyle modification that increases glucose-like transporters.
Is high LDL a concern? If so, what are some ways to drop it over time?
You want to take this one?
High LDL can definitely be a concern. I do think the universal high LDL is bad isn't really a good message to be sending. As we learn, I forgot the exact studies. I may misword this a little bit, but there were studies that were showing with all-cause mortality with LDL, and like the lowest LDL group actually had higher mortality than mid-levels of LDL.
God.
I honestly I saw someone post that.
Sydney Heart Diet Study or Minnesota Coronary Experiment, basically what the saturated fat group of people like to say for the PUFA argument, that basically PUFAs and lower LDL was correlated with higher mortality. And there's some interplay with that data. Oh, God, I have to look at the data, but if I'm not mistaken, the reason that the PUFA supporting side of people say that the data is basically worthless is because at the time they were also eating trans fat, and those are the fats most associated with mortality and cardiovascular disease. But I might be wrong on this. It might be another study. Those are the two most popular ones that I know for sure. But keep going.
I think something to keep in mind too is Like example, my wife is like genetic freak, super lean, super muscular. Like we work out every day together. We do the same workouts, bodybuilder workouts, like every day. Really good shape. We take care of our bodies. She went to get blood work done, and a nurse called her and said, "Hey, your LDL is high. You should consider dieting and doing exercise." And clearly, that wasn't the nurse that pulled the blood out of her like striated, veiny, muscular arm and just saying that, because she has genetic LDL runs high in her family, but she is pretty much as physically fit as you could possibly be, and her LDL is it is high. So is that an immediate concern for us right now? Is that actually contributing to artery plaque formation? It's hard to say, because her basal amount of LDL will be higher by how she produces it. I don't think that's as immediate of a concern. Later, when we're older, there are things that can help lower the LDL. But what do you think, Pedro? Like, because you are having the higher production, but are the arteries also used? The artery cells, are they used to that higher production of LDL? If that makes sense.
The problem comes down to the pathophysiology of how LDL contributes to heart disease and plaque development in the first place. And it's by far not the only contributor involved in plaque formation. In fact, what I've seen with recent studies are immunogenic factors are more at play here than just nutritional factors, specifically chemokine receptors and genes involved in macrophage polarization. None of that is talked about in the context of heart disease, because what you look at histologically is the fatty deposits and the crystallization of cholesterol. And so they look at primarily the lipoproteins that transport it and not the other factors like immune cells. I'm also somebody that has LDL on a higher level. That's my baseline. It's always been like 180 to 200, even when I was basically not eating eggs and meat at one moment in time when I tried a vegan diet for a couple of weeks a while back. I was still somebody that had pretty high LDL, and I still am. That is my baseline. And because of that, and because I don't think the pathology is fully understood, and because I think it's so hyper-focused, I don't think LDL as a marker is necessarily the only thing you should be looking at in relation to heart disease. It's very important nonetheless. And there are studies that do suggest, hey, the causality here is likely. And I don't deny that, but it's by far not the only factor.
Yeah. It also depends too. Yeah, again, how unhealthy are you? You know what I mean? Like if you're actually if that LDL is truly just from being sedentary, eating the wrong foods, and not improving other aspects of health markers. Like you consistently work out, you're going to have a lower resting heart rate. You're going to have stronger blood vessels. You're going to have the immune system to help break these things down. Like it will be better. So universally across the board, saying that, yeah. I don't really think that's true.
Again, it comes down to the population. Most studies, what are the population that they're looking at? People with high LDL, people with low LDL, are they generally healthy? What's their dietary intake like? Do they have good lifestyle habits? How much can researchers consider in a clinical study to actually translate to actual human beings like ourselves? It's ridiculous to just take empirical data and say, well, because empiricism says this, we're going to go and suggest that everybody should have their LDL below like 80, whatever the units of measurements are.
Yeah, and that goes back to what we said before. In an ideal world, in Dr. Tyler Panzner's ideal world, Pedro's ideal world, you do whole exome sequencing for every participant in any clinical study. And you map all the responses to figure out what gene. Imagine you could do that for all these studies. You know, how much more, obviously that's not remotely feasible time, effort, financially wise. You know, they barely get enough funding to begin with. But imagine if doing the AI to figure out which genes are more common out. You're doing these GWAS studies basically, but polygenic across the board within every other study to really see these people that do have the higher LDL, and then maybe when they follow up actually have poor health outcomes. What are the genetic differences there?
Yep, like I mentioned, one of them immune researchers in Stanford, what they did is they took a program that they made with artificial intelligence to take a bunch of participants, assess their genes, assess their markers of inflammation. And what they found is that the people at highest risk for cardiovascular disease were not associated specifically with LDL, but chemokine activity and pro-inflammation based off of immune factors rather than LDL itself. And it only happens also after a certain age. It's like as soon as people hit 55, there's proteomic differences in their blood plasma that change, all of a sudden increasing the risk of heart disease. And most patients that get heart disease are older.
Yeah. Yeah. And if you have that genetically higher LDL baseline, but your inflammation's low, how much does it really matter then? You know, I wouldn't be surprised if the immune cell, you know, the activation of the, like you said, the chemokines, the activation of the pro-inflammatory pathways would, this is a crude, you know, crude metaphor, I guess, but like you could have the inflammation kind of helping to glue, you know, these plaques onto the artery walls, if that makes sense. Like the inflammation coexisting with the high LDL is much more of the issue than just the LDL itself. Like what's the actual mechanism? How is it? Could it be the chemokine receptors on these endothelial cells are making them express glycoproteins that are helping to let cholesterol and stuff adhere to it? I don't think any of that data's out, right? I don't think anyone's looked at that.
They wouldn't even look at that because they look at LDL receptors in specific.
Gotcha.
So I highly, so even if the way you're describing the physiology is possible, but they don't even consider that.
Like I know inflammation, one of the papers for my PhD was doing inflammation with monocyte like trafficking across the body and how you have the different adhesion receptors on the endothelial cells in the bloodstream with the inflammation. So with or, it was in the context of with or without HIV infection. So with an HIV infection, you're having that, granted it might be a slightly different inflammatory pathway than, I don't know, just leaky gut, but maybe not. But that does allow I'm sure you probably don't have to rolling with the monocytes. They like roll along the vasculature and slowly stick, and then they do the what is it? The
transcytosis.
Exactly. Transcytosis. Yeah.
And integrins, the proteins that help them well.
Yep. Integrins, cadherins. Yep.
Very, very important stuff to consider too with heart disease, because the calcium-based it's calcium CA modulin, I think it's a protein. Do you know what I'm talking about?
Yeah.
Modulin basically is one of the factors most strongly associated with many cardiovascular diseases. How often do you hear about that? All you hear about is LDL and cholesterol.
Yeah.
That's what. And when you focus on inflammation too in these studies, they're not looking at CRP. CRP is like the last thing you look at when inflammation is already really bad, and then it'll be elevated. Most of these markers are more cytokine based for communication purposes.
Yeah. And there's no real I mean, you can do cytokine profiling, but that's not direct to consumer. That's incredibly expensive R&D laboratory technique. You know what I mean? But it would be great. I keep saying I want to have a flow cytometry machine in my man cave whenever flow cytometry, guys, is how you can literally get a sample of blood. I did this with mice during my PhD. You give them cancer, and you don't have to have cancer, but like you get a blood sample and you stain all the different subpopulations of immune cells. There are dozens of different subpopulations of immune cell subsets. They have this receptor, but not this one, but this one, but not that one. So it behaves a little differently. And you can map out exactly the immune profile, all the immune cells in your blood, what different activation states are there. And ideally, being able to have that in your basement, which no one fucking has, by the way. Hopefully I could have to have the success to have that in my basement. But like being able to eat any food, I can go eat those fucking almonds that always set me off, or those onions from the sulfate from my CBS mutation. And I can literally get some blood and see which immune cells are responding to this. Now, sorry, getting ahead of myself there. But
I was going to say, after the PhD arc, you and I could probably build one, man. Because if I do molecular engineering, I'll learn how to do all this stuff. Anyways, how to build a real flow cytometer. And I've seen the outline. It's not that crazy. It's just you need a lot of high tech tools and a lot of money.
Yeah.
Somebody asked, in terms of education pathway, they have a bachelor's in biochemistry. They have a manual therapy degree as well, and they're looking to potentially become a ND, naturopathic doctor. But they're open to other colleges, certification programs, reading, studying, etc. What would we recommend? Do you want to take this one?
That's hard just because I'm more on the PhD scientist side of stuff. I just know that if you get good enough at what you do, like you kind of like hop around, kind of like what I did. I was trained to pipette cells and inject mice with cancer and analyze immune cells. That was my training was, but you have enough of like my PhD wasn't in genetics. Like I took graduate genetics, I took graduate molecular biology of the gene. but I did CRISPR in my lab, but it wasn't heavy genetics focus, but like I just used the framework that my education gave me and taught some of myself some of that other stuff. And I think as long as, obviously if you're clinically, I won't be able to, I can't clinically prescribe anything. I'm not a pharmacist. I'm not an actual practitioner, you know, but utilizing that, I kind of found my own niche where I can still help people using these over-the-counter stuff and using my expertise to find out what really works best. Wish I could answer that question better. I think it also depends. I'm very, Pedro as well, very like creative, outside of the box thinking. And that's what PhD really trains you to do. The philosophy of how to find out new things. Nothing against naturopaths, you know, nothing against that, but that's more so, I believe at least the training will be more so like, I'm trying to get a word for this, like actual lectures and training on how to apply that, but not as much the training. Not that they don't train you to critically think. I don't want to come across like I'm bashing naturopaths by any means. Just a different type of training. PhDs are focused entirely on innovation. You have to prove something that no one else has ever proven before. And not even a fake little lab experiment. You got to literally learn this stuff and find out something brand new and prove it against peer review and get it published. So if you're someone that's always thinking differently than other people are or whatever, maybe that could be better. But it also depends if you like research or not, because especially with these health related PhD sciences, you're going to be doing research. There's really no way around that.
I strongly agree with what you mentioned. If I can discuss more the clinical side, to contrast that, as a clinician, whether you're naturopathic, medical doctor, chiropractor, whatever, you learn for the first 2 years your baseline requirements, like higher anatomy, physiology, higher pharmacology, but you never get deep enough unless you decide to do research with it, which is very difficult to do with a medical degree. You never get deep enough to really know why things are the way they are. You kind of just, you're applying because you're trying to take care of patients. If you're interested in helping people as a clinician and patient interaction based degree, that's great. But if you're looking to actually do research and understand further about why certain patients have the issues that they do, well, then you're more than likely better off doing a PhD.
If you're asking the why constantly, that could seem more that way. And it's ironic too. I'd me personally, I didn't want to do a PharmD or MD because I didn't want to have to deal with interacting with people. And I realized, though, I'm a very social person. It wasn't, and now here I am doing this holistic genetic health coaching, interacting with people. So it's like, I realized it wasn't even so much I don't want to be interacting with people. I love helping people, but it was more so in the context. I didn't want to be interacting with people just saying, here's your medication in retail pharmacy, or here's your medication for this or that. I wanted to actually figure out why, how can I make this better for this individual, whatever I'm giving them, and why is that the case? And as I went down this rabbit hole, that's led me down the genetics and how you can use these natural things, personalize to somebody and get really good effects if it's hyper-personalized to what their cells need.
One of the questions here was from my buddy Chris, and he's mentioning relation between Epstein-Barr virus, high amounts of B12, folate, and zinc. So I'm assuming the question is like, because he's dealing with a couple of things related to Epstein-Barr virus right now. I'm not going to explain because that's his information. But if we have Epstein-Barr virus infection, what are the interplays with higher doses of vitamin B12, B9, and also zinc? Is it a good thing? Is it a bad thing? You want to take this one?
I mean, I'm definitely not an EBV guy, but just from the genetics point of view, I think people look at the blood doses, blood tests for different vitamins and stuff, and they're useful, definitely. Like, there's a lot of clinical data around it, but at the end of the day, that's measuring the amount of a nutrient or vitamin that's in your blood that's not actually inside of your cells. And that's why they have things like red blood cell magnesium tests, where they get the red blood cells alone, lyse those open and see how much is in that. But even again, we were talking about before how different cell types preferentially need, require, and receive different amounts of nutrients. So like, I know personally, I look at mutations that affect the B12 absorption in the gut, the B12 transport. So the proteins themselves that carry B12 through your body, they've actually done some clinical studies showing that when these proteins are mutated that can't transport B12 into your cells as well, some people have lower B12 levels in the blood. Some people actually also have higher B12 levels. And that's because it's staying in the blood and not getting into the cells where it really matters. So I've worked with some clients that have a lot of mutations there, and they take all this B12. It's not really doing that much. And I know lithium is something that needs to be cycled. You got to use it carefully, make sure it's the right person. But that is a critical cofactor for vitamin B12 transport inside of your cells. So they start taking lithium, and then they're like, oh my God, this is what B12 is supposed to feel like, because they needed that boost to kind of shove the B12 into there. So regarding the EBV context itself, that's more of a Pedro thing. He'll take that after me. But regarding the B12 side, making sure that B12 you're taking is actually absorbed. People with a lot of homozygous mutations and gut absorption of B12 and can absorb it through dietary or supplements, IV infusions may be good for them. But now the issue with that, though, is that I've really only seen either cyano B12 or methyl B12 for these IV infusions. I'm not aware of anyone doing a denosol or hydroxyl B12 infusion. So I've had clients that have mutations in absorbing B12, so they need to get an infusion. But they also have a COMT mutation. As we mentioned, that methyl infusion will be really bad anxiety for them. And I've had a couple of clients come to me and said they got a methyl B12 infusion and they lost their shit for like a whole day because you're just giving that exact clashing nutrient to them. So if anyone's doing in the the infusion business, start doing adenosine hydroxyl B12. I think that's going to be a huge market as more data comes out and more people are aware of this. But yeah, you take it with the EBV side of it.
Well, first I want to mention that's actually really interesting about lithium being a factor for transport of B12. I had not heard of that before. I'm looking at a paper on this right now, so I'm going to look into this after. But in terms of Epstein-Barr virus, it's a tricky business because EBV, the reason why people have symptoms of reactivated or chronic EBV in the first place is it goes into its latency stage inside of immune cells, particularly, and primarily B cells. And once it goes inside of B cells and you have a stressor or an immunogenic antigen and it activates the immune system in a pro-inflammatory way, the immune system is going to start activating all of its processes. Metabolism is going to go up, and so will the translation of Epstein-Barr virus related non-specific proteins, NSPs. And so you'll make them, but you won't make the full virions in high quantities. It'll be negative on blood tests, but you'll have antibodies to the particular proteins causing the symptoms. If you're taking high doses of these B vitamins, you are supporting a higher metabolic rate that might worsen the condition. And so it depends on the person and the situation. However, zinc is one really important compound that does elicit extremely good responses with pretty much any viral infection. And the way that zinc is doing this is by inhibiting the actual translation with the enzyme called RNA-dependent RNA polymerase, RDRP. This enzyme is necessary for the Epstein-Barr virus to be produced in the first place. What zinc does is it binds to different sites of the enzyme and shuts it off from ever reading RNA. And so the virus never gets produced. So high doses of zinc for a period of time, as long as you're not getting into overdose ranges, really seems to have a great effect with all kinds of viral conditions.
I'm going to run to the bathroom real quick.
For it. All right, so I'm just going to answer questions now by myself. Let's see. Let me go back on this. Does the type of zinc make a difference? I think, yeah, it does in a small amount. All these ions like magnesium, zinc, calcium, and so on, the forms that they take outside of the body are important for transport and for utilization by cells. But inevitably, all of them become ions: zinc ion, calcium ion, magnesium ion. So the forms don't matter as much. It's like a small contributor because inevitably they're going to be liberated anyways. So I hope that answers both questions. And there are obviously ways to help people. Foundations, yeah. All right. There are definitely genetic factors making people hypersensitive to glutamate, and glutamate in general will promote excitotoxicity in the brains, so it's a possibility. All right. Hair analysis test results. Yeah, I mean, they're useful, and especially with particular heavy metals that are toxic. Aluminum is a big one. I definitely think that could be useful to diagnose high aluminum levels in people. Let's see. If you were to recommend one type of magnesium to take, what would it be? I would recommend magnesium glycinate for helping with sleep. Quercetin does help zinc absorption. It's in ionophore.
Quercetin also spikes. Quercetin also, and here I am. You know me. I got to chime in on this. We mentioned the COMT enzyme and then the MAO enzyme. I didn't mention that yet, but that's like these are the two enzymes that break down adrenaline and dopamine in your brain. It's one of these two, and quercetin blocks both of them very, very strongly, and it has a very long half-life. So be wary. That's one of the top supplements for people that have this anxiety. I see a lot of people come to me, they're trying to fix their mast cells or taking quercetin, and it's causing their anxiety. And yeah, just be wary of that. Anything that, you know, affects those enzymes, I talk about a lot because I'm so sensitive to them, and I've had a lot of really shitty days by taking some of those supplements in normal amounts. That does not mean they're bad or toxic for anybody. They're still very, very good at what they do. It's just if you start taking stuff and feel really, really anxious, you know, it's all about pros versus cons for you.
Yeah, I agree. Let's see. So mineral balancing using that's my buddy, my client actually, Yost. Mineral balancing using HTMA to rid heavy metals. Yeah, Tyler, you seen that page? I don't know who the guy is. I think you follow them with the mineral balance, how minerals interplay with biology in general.
Yeah.
How do you feel about that stuff? Because I'm kind of, you know, especially doing inorganic chemistry, it's all about metals. It's almost like as soon as you try to over supplement with one specific metal to get rid of the other, and you try to chelate bad metals at the same time, it's counteracting the therapy in the first place because the chelation is non-specific.
Yeah, I'm honestly not too. That's one of the things this next year I really do want to learn more about that, because just from a cell biology, cell behavior, that just seems like so hugely important with things. I've had some clients that use HTMA, that bring HTMA to me. I'm not well versed to give expert advice on that yet, but we will look and see. Oh, you know, we have mutations in copper transporters, and what do you know? Your copper is very low or your copper is very high, et cetera. So there is a lot of overlap there. So I must say, at least regarding the tests, tell the right stuff. Regarding the chelation stuff, I didn't even know whether chelation is specific or not. I'm just not one of those things I want to learn more about, but I wouldn't want to give an uneducated opinion on that.
Yeah.
At least with this level, I really don't know too much about them. I do know people are getting a lot of good results. I've heard of, especially with like things like autism, that can help a lot with a lot of mineral imbalancing. I don't doubt the validity, but again, from what you said, if the chelation is not specific, then you should probably do like a chelate then replace rather than do it both at the same time.
Right. One thing I'll mention, Yost, in regards to HTMA analysis is usually the biggest concerns are like mercury, thallium, usually from vegetables, funny enough, and then aluminum. And in that case, the things that you're doing at the moment, like with increasing specific vegetable intake, sulfur-rich vegetables in particular, they'll already be supporting chelation therapy. Fiber is really a huge component of heavy metal detoxification. Without it, you're going to accumulate it in fatty tissue. That's the one thing I'll say regarding
Interesting. So I wonder, so could then carnivore diets technically make heavy metals worse?
What's interesting about that is the actual fat intake of people that are pursuing carnivore diets, ketogenic diets, and the increase in cell horrible. I messed up there.
Don't cancel them, everybody.
Yeah, we're going to keep that. Autophagocytic process. The amounts of metal that accumulates in cell is much less. And also you reduce the amount of glycogen and water that's stored in cells. And so it helps with eliminating the metals even more. In consequence, even carnivore diets help with this.
Interesting. But I was mentioning mainly because you mentioned the fiber aspect, which the carnivore. So you're saying there is some compensatory mechanism that replaces the lack of fiber then regarding to the heavy metals.
Yeah, exactly.
Gotcha.
And because they're already partly increasing fat intake and metals have an affinity for fatty tissue, it's likely that the higher fat intake basically coaxes it out of the body. Let's see. Other questions. Let me go to the question box.
Someone did ask regarding the quercetin or NAC for the adrenaline. NAC, I was referring to quercetin. NAC, I don't know if you were here earlier. Personally, I don't like how it makes me feel mentally, but it doesn't raise my adrenaline and make me anxious like the quercetin does. I just don't like how my thoughts work. Not that they're depressed or anything. Like it's not. If you want to try NAC, try NAC. Is what I'm saying. If you want to try quercetin, honestly. But I'm just saying, be aware if you feel overstimulated, it could be due to something like quercetin, but not so much the NAC.
Matt's got a couple of questions. Matt, what am I drinking? It was jasmine tea. And then the other question he asked was, let's see, thoughts on farmer versus hunter genetics. And this is actually something I've looked into regarding evolutionary biology, but do you want to have a take on this?
Yeah, I know that there are, I forgot the exact gene names. I think it's with glucose processing that are called the hunter versus gather variants. And just mean you do better on higher fat animal stuff, lower carb because you're consuming less plants, less berries, etc., and then vice versa. I'm pretty sure I'm heterozygous for that, so it's like I'm middle of the road for that. Again, I do think that evolution genetically selects for a set of genes that makes you most able to digest, process, absorb, and utilize the nutrients on whatever food sources are around you. But then I guess if you go to the hunter side of it, how far are you having to hunt to find these animals? You know, right. I think the hunter lifestyle is what drove them, I think, from Africa to Europe through, you know, the land bridge that was there over to North America. Yeah. So how much is that really displacing them from where they were genetically, geographically supposed to stay? But then again, I guess if they're consuming mostly animals and chasing them, it may not matter as much.
I agree. The funny thing is most of the hunter genetics are the favorable genotypes that people look for in a partner. It's inevitably you know where I'm going with this, right? Looks matching, basically.
I thought you were going to say something like the MHC type stuff with the scent while you're saying like mate preferentiation.
Mate preference, yeah, but in all ways, not just like scent is a big one because of pheromones, but in general, for example, the thicker eyebrow region here, the thicker it is, the more that it's associated with a particularly robust male.
The caveman shit. I have that right here too. Certain pictures in shadow, and I'm like, yo, I look weird.
If your jawline is recessed because of nutrient deficiencies, which are very common with old farmers,
and oxygen too, I think, with the nose breathing, mouth breathing stuff too.
Right. Yep. It all affects brain processes as well because you're pushing on the spinal cord in a certain way. The brain is not oriented in its proper place. The palate makes a big difference in how your brain functions and you respire as well. All of those traits were favored by hunter-based genetics. And one of the reasons why is because they were less nutrient deficient because they were basically getting all their nutrients from certain sources of food like meat. In comparison to farmers, they didn't know a lot of the farming practices and important things to consider now that they were unaware of back then, such as the phytic acid anti-nutrients that chelate certain minerals. And so they were basically promoting a state of nutrient deficiency for a long period of time. And it's one of the reasons why hunter-gatherers, once they got to these and geographic locations in general where there were farmers, they basically pillaged the entire place, raped the women, and then promoted their own genotype by having offsprings. Wow. It's a harsh truth to come to, but one that's very it's rooted in history. It's cemented in history in a sense. You can't deny what has happened evolutionarily in the promotion of this genotype based purely on conquest.
No, that's a good point you brought up regarding you're growing the food, you're surviving, you're gathering. You don't have to hunt now to go. You don't have to go anywhere, so you can be more sedentary and build like an actual civilization, less nomadic. But at the same point, you're not knowing about the phytic acid, this or that. Like you're making your lifestyle feel easier in the moment, but at what cost down the road if you're chelating all these minerals, et cetera, anti-nutrients and stuff. Really interesting.
Evolutionary biology, man. Let's see what other questions.
We already did the blood type, but everyone asking, we're definitely going to save this, and we did touch on the blood type earlier.
Let's see. Thoughts on the best diet for autoimmunity? Keto versus goodbye lupus. I'm assuming that's a diet, which is high in omega-3s via seeds and raw cruciferous vegetables with some fruit only. What do you think?
I know that there are genes associated with like definitely gluten sensitivity, definitely oxalate sensitivity, emerging data about lectin sensitivity. I haven't seen too much data on the nightshade sensitivity, but again, you follow the trend. What aggravates your gut, what molecules in these foods aggravate your gut more, are based on the genes, the host genes ultimately. And for the autoimmunity, at least, let me know what you think about this. I think basically the inflammation is either coming from the gut or an open wound. Otherwise, what else is triggering that initial? It can linger for longer, you know, from pro-inflammatory autoimmunity antibodies. But initially, I feel like a lot of that is stemming from the gut, or mostly that, versus an open wound. But I think it does. Any disease has many different flavors. So any autoimmune, even two people with lupus, it's like a syndrome because there's no one exact type. Some people, I believe genetics are at play for every single disease there is. What percentage? That depends. Some people may have more of a lifestyle type thing or this or that, but autoimmunity with lupus is a good example. Some people may be having more inflammation, gut inflammation from the oxalates they're eating. They can have mutations in how they break down these oxalates. And one telltale sign to tell if you're sensitive to oxalates, do kidney stones run in your family? Kidney stones are primarily made from these oxalate crystals that form over time. But it's also been shown that before you form, that's the end stage. Before that happens, you have a slow burn where you're affecting the immune cells in your body. You're causing inflammation in your body before you start forming kidney stones. So if you're getting kidney stones, you've been having inflammation prior to that. Whether or not you could perceive that or not, that just depends on the individual. But I definitely think the best diet just depends on which stuff is, I guess I'm talking from the food and gut inflammation perspective. Then there's the whole, what nutrients are in the food, but that could be a whole other separate live. What do you think?
Yeah, specifically bouncing off of starting from the gut perspective, it's clear that the gut is playing such a large role in people's diseases because it only takes a couple of days of eliminating all foods except a couple, like say meat and water, for example, with carnivore diets, before people's symptoms completely go away.
Yep.
So if that's the case, obviously there's a gut interplay here. We know that the gut is the major site, one of the major sites of immune maturation. The immune cells, not only innate, but adaptive immune cells do go there so that they can interplay with all of the antigens that come across the lumen of the gut so that they can mature and know what's good and what's bad. So there's obviously a huge immunological role if your gut is unhealthy. And then in regards to lupus in specific, the oxalic acid and oxalates in general, one of the major contributors are yeast. So if you have yeast infections in the gut, that'll bring it up. The final thing I would say is there is one really interesting marker with lupus that I think is indicative of viral concerns, and that's free floating DNA in serum. You just have a higher load of DNA in serum.
Cell-free DNA.
Which, in my opinion, is mostly viral mediated. So it's likely that there's contributors.
But is it host-free DNA? So like cells are being lysed, or it's like not actual viral genomes?
No,
host DNA.
It's mostly host DNA, but through mechanisms that seem to be viral mediated from what I have read.
Gotcha. Someone asked 23andMe good enough for self-decode? Yeah, you can check their website. Most of the main DNA companies they accept it from, and if it's not on that list, you can check out their website. They'll show you shoot them an email, and they'll basically let you know if it's good enough or not. But pretty much all the major ones, you could put that in. I've never been sequenced through self-decode. I put my 23andMe in, and I got all my data.
Somebody asked, thoughts on curing acne? Mine isn't horrible, but I get some on my face, chest, and upper back consistently.
I think, again, it depends. A lot of people think in regards to symptoms or disease or health ailment and like what to do to fix that. Well, it may not be the best wording. Like acne, for example. What's the best thing to take for acne? What's the best thing to take for X or Y or Z? It's not just symptoms and supplements or drugs. It's in between that are the cellular pathways that are dictating the cell behavior. So clearly, certain pathways are dysregulated in the skin cells. Otherwise, they wouldn't be recruiting the immune cells to be causing this inflammation. So it depends. Again, all different ailments have different flavors going on. Histamine, elevated histamine has been highly linked to different types of acne, hives, eczema. Some people, it's the joints, this or that. But histamine could possibly be a root cause for you. It may not. It may be LPS from your leaky gut that's causing that. It may be mutations in gamma-linolenic acid deficiency. That's been linked to joint health, so joint pain, as well as some skin inflammation. That's why things like borage oil or black seed oil, people recommend that a lot for skin issues. And that's not just because this works for all skin issues. It works because this supplies your body, your cells, with gamma-linolenic acid, which then restores that deficiency and kind of quells and lowers some of that inflammation. So it really just depends on what flavor you have going on to really get to the root cause there. I mean, or you can go down the list, different mechanisms. Like I mentioned, histamine type stuff, lowering histamine, getting food allergy testing, that may be one thing. Or as I mentioned, the GLA from borage seed oil or from black seed oil, that may be efficacious as well. Or I've seen zinc deficiencies causing that as well. It depends on what the cells are actually lacking or have too much on. And that's different for every individual. I don't even know how many flavors of acne there are. You know, I think of it as like a painting. Like you have a little more of color red of this pathway, a little more of blue, too little of green. Everyone's all a unique canvas of that. And going back to the clinical data, that's why when you see no change, no effect, that was such a good point you brought up, Pedro. What about the 30% of the people that did see an effect? Clearly it worked for some people. So why are we saying there's no significant effect on human health? It did have a good effect. There's no way all of those people were flukes. I get one outlier, but 30% of the population in that group is not an outlier. That's a subset of individuals that I believe have specific genetic mutation combinations.
I will mention in regards to acne, you mentioned mechanisms that you can rule out. One of them that's a huge contributor, and I think most people are facing this, it's insulin resistance and also a inability to oxidize fats effectively.
So there we go. That's two more flavors. I think I mentioned 2 or 3, and we're up to 6 right now. You know, different drive. You can have multiple of them in different amounts. It just depends on the person.
Yes, one guy said blastocystis hominis, or I'm not sure if that's the exact name, can be an underlying cause of acne. A lot of microorganisms can. Yeah. One of the major ones is propionic bacteria. The reason why is because, again, with fat oxidation, if you're not burning those fats effectively in cells and the sebaceous glands in the skin are producing more fats, they're going to take that and break it down for their own purposes. What they produce in return are pro-inflammatory fats that are broken down from the original fats you produce that promote skin inflammation, and thus acne occurs. And insulin resistance is a big driver of that.
Yeah. Yeah, the micro part is huge as well. You know, like obviously, whether it's the gut microbes communicating or going through the leaky gut, getting up there, or literally just being on your face as well. And I wonder if the insulin resistance also affects immune cell dynamics as well. So I'm sure they're kind of interrelated there. I mean, the one thing I'm realizing is everything affects everything. Everything's interrelated. That's why,
One of the questions was by somebody who their grandfather got can't mention on the live because we'll get banned, and I think passed away from it. And then she's asking in regards to recovery from atrial fibrillation. Anything on that? Why we think that causes basically heart failure or sudden cardiac events?
I do think, at least I know, I'm looking a lot into interleukin 17, and I made a post, a story thing a little while ago, how IL-17, it regulates autoimmunity. And basically any autoimmune condition, arthritis, lupus, rheumatoid arthritis, basically all of them, psoriasis, there's all literature showing that higher IL-17 is responsible for that, including lingering viral infections. So including long, that viral infection, that's been shown, regular version of that has been shown to elevate IL-17. And elevated IL-17 has been shown to cause persistent viral infections. Even though they haven't proven long, is really like due to IL-17, you can read between the lines there. I'm not sure regarding that specifically, but I don't know. You know what? I'm going to look right now. What was it called? Atrial
fibrillation.
Fibrillation. I'm going to look up right now and see if that's actually interconnected there.
IL-17.
Yeah.
I would imagine it is, since the renin, aldosterone, angiotensin system, RAAS activation is part of it.
So I'm going to look for that. That makes more sense for a link there.
In terms of what I think about why people are suddenly just collapsing, if it's related to this, which obviously I think there's a major influence from what. It seems to me to be a nerve issue rather than a cardiac issue, like pericarditis, myocarditis, anything like that. It's almost like the atrioventricular, which ones are the sinoatrial node and atrioventricular node, the ones that conduct the heart in the first place, are being affected from persistent inflammation, whether it's due to lingering RNA, whether it's due to lingering immune subtypes, immune communication. Like I mentioned in the beginning of this podcast, immune signaling is a major portion of cardiovascular disease, and it's not talked about enough. And it could be that there's persistent immune signaling around those conducting areas, the nerve bundles in general around the heart, that then cause sudden cardiac events that are life-threatening. So that's what I think is going on in regards to that. And then atrial fibrillation, there are a lot of contributors, but increased platelet aggregation because of thick blood, viscosity in the blood is a major factor. Increased sodium intake and not enough potassium, increased angiotensin level because of higher body fat. Those all affect atrial fibrillation.
I mean, and then of course I have to chime in the mutations that affect the angiotensin system. I know that there are some mutations in, I think it might be MHC proteins, so immune recognition, that are linked to adverse effects of and again, it's a mutation in the renin-angiotensin system,
which is huge.
It indirectly would affect possible side effects to that. So yeah. And then how much do you think would be the spike protein as well, though?
I don't think
that's pretty fleeting. Like it stays longer than they thought, but that wouldn't be this. At this rate in the game, it would be. Yeah. I think you mentioned too. What were you saying? The late stage IgG, the
IgG class switching.
Yeah, the class switching. I remember you mentioned that to me. We kind of spoke about this in private.
Yeah. There's not that much information on it. I don't think it's the major contributor. I think it's a consequence, but not the cause of what's happening with like sudden. Yeah, it's definitely a factor though. And a lot of people have denied that. I've seen videos from more mainstream practitioners and stuff saying that IgG class switching tends to occur even with normal infection. That is not what the paper said. If you take a moment to go through the paper, it doesn't occur with most viral or bacterial infections. It tends to occur predominantly with what they're using at the moment with the mRNA-based vaccinations. Even old school vaccinations don't cause this kind of extreme IgG class switching after a couple of doses of it. So interesting. No idea though. Let's see other questions. How long do you want to keep this going? Because we've been at it for 2 hours.
Yeah, I could go for another 15. Then I got to eat, get ready for a client call.
No problem.
So
let's do like two or three more questions then. Let's see.
I got to host the next one. I'm dying to know what this question box looks like.
It's not that much actually. We answered most of it. Yeah. I'm just going off of not the question box, but the actual chat as well.
Gotcha. Oh, you're saying you have to put it in a special area, so not everyone that's in the comments will go to the chat box.
What are good ways to help increase the conversion of glutamate into GABA, in addition to just vitamin B6?
Glutamate into GABA, in addition to B6. That is one of the major cofactors for that conversion there. But what I've personally been using with some clients that are having issues with that conversion, the glutamate to GABA, right?
Yeah.
So have you ever heard of picalamon? Picalamon? It's niacin conjugated to GABA, and it lets it cross the blood-brain barrier. So one of the main downsides of GABA supplementation is that it may get into the brain, but you pump it out just as quick. I think you can still get some stimulation from the gut enteric neurons and get some calming. Like I take GABA and it does calm me down. So clearly it is doing something, but it is somewhat fleeting. So pairing it with something like a lemon balm. So rather than at a certain rate, if people have mutations, I think it might be in the GAD enzymes, I believe, that do that conversion. I believe so. But if you have so many mutations in that pathway, you can only give so many cofactors before you reach the maximum rate of conversion there. And if that's still not enough and there's still issues going on, then the next best route would be to figure out ways to boost up GABA. It's about the ratios that matter more so than the raw amounts. So if the glutamate's a bit high and you're having some issues, increase the GABA to kind of have that ratio there if you can't be converting it as well. But picolamone, I've seen really good effects with that because it does cross the blood-brain barrier readily versus the regular GABA.
I'm looking at it right now, and it's amphoteric, so one end positive, one end negative, or rather one end fat soluble, one end water soluble, and that could be one of the reasons why it crosses so effectively. Let's see, one more question. I think we'll call it a day. Okay, we got the questions from the question box done. So let's see, go back up here. Amyloids have been found in various areas from the thing. Oh, it's not a question. Never mind. No, nope, got that. No. Pedro, you've recently posted something on nerve regeneration in regards to brain issues. Have any help or improvement in somebody who has incomplete spinal cord injury? What do you think? Spinal cord injury.
I know that that's way different than just neurogenesis, if I'm not mistaken. Yeah. Like they're definitely a different degree for that. I honestly haven't. I used to follow regenerative medicine a bit more, but now I've been doing like a couple of years. It used to be one of my main interests, but I honestly haven't kept up. I know they have been getting promising results in mouse models, but I'd be lying if I told you exactly what would be what they were using for that. I know there's some peptides that can help with regeneration. Like there's like this BPC 157. Does that affect neural tissues at all? I know it's really good for gut issues, for ligament issues, connective tissue issues.
I wouldn't doubt if it did. And new peptides is the other one that I can think of that does help with nerve regeneration. Many peptides have really good effects. Steroids as well, not necessarily like pure testosterone, but most pro-hormones and most anabolic-like compounds do promote nerve regeneration. And the thing is, yeah, the spinal cord, it's such a tough one to heal properly. It's an incomplete spinal cord injury, though. So it may heal. It depends on, I believe, what the differentiating factor in spinal cord regeneration is how deep it goes. It cuts into the spinal cord. If it's like the outer layer, it's possible to still regenerate.
Yeah, I'm pretty sure you have to have a certain amount of those cell to ECM or cell to cell interactions. Like it's very hard to just have a clean tear and get that to work. If you have some of those adhesions there, I know that does have a much better prognosis, but obviously a partial tear is better than a full tear. So
I agree. Yeah, it's tricky. Not quite sure on this one.
Is BPC 157 effective orally? Yeah, I believe so. Yeah, they have a couple formulations that for that to work orally. Especially good for gut health. It can help with joint health as well, but I think the local injection for specific joint issues would be superior. But there are orally effective versions.
Want to call it a day here?
Yeah.
That was good, man. And we did a lot. And again, 2 hours felt like it was like 30 minutes.
Yeah, we blinked, it went by. Thank you, everyone, for coming in, asking questions. We're definitely going to try to do this more often. Hopefully at least once a month, maybe twice a month.
I want to
try to see with our schedules.
Some guests in too. Maybe some of our buddies can come in and join us.
Yeah. I forgot you could do the multi-wave live as well. Awesome. And yeah, I sent you the see if you could try to, I guess, end the live and then open the email and download the thing. If the thumbnail's botched, it is what it is. We'll be fine. But yeah. All right, my man. Great chatting with you as per usual. Hopefully you guys got a lot of value out of this. And wishing all you guys happy health, happy life, happy everything. And until next time.
Take care. Talk to you soon.